Hello everyone, welcome back to my channel. Today we are going to be talking about topic 8a, the nervous system. So without any further ado, let's begin. Okay, so first slide is type and function of the neuron. Neurons are specialized cells of the nervous system which carry electrical impulses around the body. So a bundle of neurons such as that is known as a nerve. Now, what is the structure of a neuron? The basic structure of all the neurons contains a cell body which has all the nucleus, mitochondria, rough endoplasmic reticulum, ribosomes, where protein synthesis occurs, and other organelles. The next is dendrites. Dendrites are thin extensions from the cell body that branches out and connects to the neighboring nerve cells. So this allows impulses to pass from one nerve cell to the other, enabling a network for easy communication. Dendrites are mainly for allowing impulses to transfer from one nerve cell to the other, and this establishes an easy communication. What are axons? Axons are long and thin nerve fibers which carries nerve impulses. So axons are the actual fibers which carries the nerve impulses. Next is myelinated nerve fiber. Now, nerve fiber is only occurring in some neurons, but not all the neurons. So, myelin sheath is made up of specialized cells known as swan cells, which wrap around themselves around the axon. Now, this helps to speed up the rate of nerve transmissions as well as protecting the nerves from the damages. Then we have the nodes of Ranvier. Gaps exist between these swan cells, which is called nodes of Ranvier. This is where electrical impulses in myelinated cells do not travel down the whole axon, but rather jump from one node to another. Now, electrical impulses does not travel down the whole axon of a neuron, but rather they jump from one node to another, and these are called the nodes of Ranvier. The next one is non-myelinated nerve. As we have discussed before, that not every neuron contains myelinated nerve fiber. The non-myelinated nerve fiber is the one where the axon is not insulated by the swan cells, and hence impulses travel more slowly as it moves down the entire length of the axon. Okay, moving on to the next slide. Structure and function of sensory relay and motor neurons. So there are three types of neurons. First is sensory, second is relay, the third one is motor neurons. Now, in the picture, besides the table, here is a diagram of the three types of neuron with their structures labeled accordingly. Let's learn more about the sensory neurons. Sensory neurons have long dendrites or dendrites which collect impulses from receptors. These, their cell bodies are located outside of the spinal cord. They have short axons transmitting impulses to the CNS. They carry impulses from sensory neurons, for example, the skin, eyes, and ears, to the spinal cord and the brain. Second, we have relay neurons. Relay neurons have short dendrites and axons. They are completely within the central nervous system and they connect sensory neurons to motor neurons. Relay neurons are important in reflex actions and processing information in the central nervous system. Third is the motor neurons. In the motor neurons, cell bodies are inside the CNS. Short dendrites and long axon extends to the effector cells, such as muscles or glands, and they carry impulses from the central nervous system to the effectors. Example, the muscles contract and or glands are secreting. Okay, now how the nervous system of organisms can cause effectors to respond to a stimulus. A stimulus, uh, for example, could be heat or pain or light, and this is detected by a sensory receptors. For example, thermoreceptors in the skin or photoreceptors in the eye. The receptors transduces the stimulus into an electrical impulse. The impulse is transmitted along a sensory neuron to the spinal cord or the brain. The sensory neuron's axon carries the impulse passing through the dorsal root of the spinal cord. In the reflex arc, the sensory neuron synapses with a relay neuron in the spinal cord. The relay neuron transmits the impulse to the motor neuron by synapsing with it in turn. For complex responses, impulses travel to the brain where they are processed before a response is generated. The motor neuron carries impulses from the CNS to an effector, for example, a muscle or gland. The impulse travels down the axon of the motor neuron to the effector. The effector carries out a response. If the effector is a muscle, it contracts, or if the effector is a gland, it secretes a hormone or an enzyme. Let's now study the detailed function of the spinal reflex arc, and this includes the gray matter and white matter of the spinal cord. The spinal reflex arc is a neural pathway that controls a rapid involuntary response to a stimulus, and it involves the spinal cord directly. It bypasses conscious regions of the brain, resulting in a quick reaction. This process begins with a receptor cell that detects a stimulus, such as a sharp object or heat, in the skin. The receptor cell generates an electrical impulse that travels along a sensory neuron towards the central nervous system. The cell body of a sensory neuron is located in the dorsal root ganglion, and the nerve fibers travel through the dorsal root. Within the spinal cord, the sensory neuron synapses with the relay neuron, also known as an interneuron. The relay neuron is located at the gray matter of the spinal cord. The relay neuron then synapses with a motor neuron. The motor neuron carries an impulse away from the CNS through the ventral root. The motor neuron transmits the impulse to an effector, which is typically a muscle or gland. In a spinal reflex, the effector is often a skeletal muscle. The effector carries out the response. For example, a muscle contracts to move a limb away from a painful stimulus. Now, we study the role of gray matter and white matter in the spinal cord. The spinal cord is made up of two types of tissues, gray matter and white matter. The gray matter is located in the central part of the spinal cord and is shaped like a butterfly or an H. It mainly consists of the cell bodies of motor neurons and relay neurons, as well as synapses between neurons. The dorsal or posterior horns of the gray matter contain relay neurons that receive impulses from sensory neuron, and the ventral anterior horns contain motor neurons that send impulses to effectors. It is the area where synapses occur between the neurons. So, if you want a more pronounced or clear wording. So it might be better if you check the Edexcel textbook. There you may have the diagram, which can be more clearer for you. So check that out. The white matter, on the other hand, surrounds the gray matter and is made up of myelinated axons. The myelin sheath gives the white matter its color. The white matter contains the nerve fibers that carry information up to the brain and down to the effectors, facilitating communication between the brain and the spinal cord. These myelinated axons allow for faster transmission of nerve impulses. It is the pathway of a spinal reflex. In a spinal reflex, like the withdrawal of a hand from a hot object, the stimulus activates receptors in the skin, which in turn initiate an impulse in the sensory neuron. This impulse travels to the spinal cord through the dorsal root. In the gray matter, the sensory neuron synapses with the relay neuron, which in turn synapses with the motor neuron. The motor neuron then carries the impulse to the effector muscles, leading to the hand's withdrawal. This entire process occurs very quickly without conscious thought. So here's the summary that of the spinal cord. Let's move on to the next slide. Now let's discuss how a nerve impulse is conducted along an axon, including changes in membrane permeability to sodium and potassium ions. Action potentials are the momentary reversals in electrical potential differences across a neuron's cell surface membrane that allow for the transmission of nerve impulses. They involve rapid movement of sodium and potassium ions across the axon membrane and cause a change in the membrane potential from about minus 70 millivolts to 30 millivolts. So, first of all, to understand how an action potential is generated, we need to understand that the first stage is a resting potential. A neuron that does not conduct a nerve impulse is in a resting phase. In the resting axon, the inside has a negative electrical potential compared to the outside, typically around minus 70 millivolts. This is due to a difference in ion concentrations, which is maintained by the sodium potassium pumps, and the membrane's greater permeability to potassium ions. The sodium potassium pump actively transports 3 sodium ions out of the axon and 2 potassium ions into the axon using ATP. Potassium ions can also move out of the axon through facilitated diffusion via potassium ion channels. Then we have stage of depolarization. When a neuron is stimulated, some sodium ion channels open and sodium begins to move into the axon down its concentration gradient. If the potential difference reaches the threshold potential around minus 55 millivolts, more voltage gated sodium channels open, causing a large influx of sodium ions. This reverses the charge across the membrane, making the inside positive and reaching around plus 30 millivolts, and this is called the depolarization. Next one is repolarization. About 1 millisecond after depolarization, the voltage gated sodium channels close. Voltage gated sodium potassium channels then sorry, voltage gated potassium channels then open, allowing potassium ions to diffuse out of the axon down their concentration gradient, which makes the inside of the axon negative again. This is called repolarization. And there is hyperpolarization, where there is a short period where the membrane potential is more negative than the resting potential, which is known as hyperpolarization. During this refractory period, the membrane is unresponsive to new stimuli. This ensures that the action potential is a discrete event and the impulse can only travel in one direction. Restoration of resting potential. The voltage gated potassium channels then close and the sodium potassium pump works to restore the resting potential of minus 70 millivolts. Once an action potential is generated, it is transmitted along the axon. The depolarization at one site causes sodium ions to diffuse along the cytoplasm to the next section of the axon, depolarizing it and causing voltage gated sodium channels to open, generating another action potential. This process repeats along the length of the axon. The action potential is considered to be an all or nothing event because it is only generated if the threshold potential is reached, and the size of the action potential does not change based on the size of the stimulus. Instead, a stronger stimulus will cause an increase in the frequency of action potential. So please understand this line very importantly, that an action potential is considered to be an all or nothing event because it is only generated if the threshold limit is reached, and the size of the action potential does not change based on the size of the stimulus. Instead, a stronger stimulus will cause an increase in the frequency of action potential. For simpler and better visual representation, there's a graph chart given here, and this chart has contained all the processes of this action potential nerve impulse. Please have a look at this. The role of myelination in saltatory conduction. Saltatory conduction is a type of nerve impulse transmission that occurs in myelinated neurons, which are neurons that have a myelin sheath surrounding their axons. The myelin sheath is made up of specialized cells called smart cells that wrap themselves around the axon, forming an insulating layer. The myelin sheath is not continuous along the entire length of the axon. There are gaps between the squamous cells called nodes of Ranvier. In myelinated neurons, depolarization and the generation of action potential can only occur at the nodes of Ranvier. This is because the myelin sheath prevents the diffusion of ions which are necessary for depolarization across the membrane in the myelinated section of the axon. So, how does saltatory conduction work? Sodium ions diffuse along the axon within the smart cells, traveling from one node of Ranvier to the next. This movement of ions within the smart cells create local currents. When the sodium ions reach the next node of Ranvier, they depolarize the membrane at that node. This depolarization triggers the opening of voltage-gated sodium channels at the node, allowing a rapid influx of sodium ions into the axon. The influx of sodium ions generates an action potential at the node. This process then repeats at the next node of Ranvier, so the action potential appears to jump from one node to the next. This jumping of the action potential is what referred to as saltatory conduction. Myelination and saltatory conduction greatly depend and increase the speed at which action potential travel along the axon. This is because the action potential does not need to travel along the entire length of the axon membrane. It only needs to be generated at the nodes of Ranvier, which are spaced relatively far apart. The structure and function of synapses in nerve impulse transmission, including the role of neurotransmitter and acetylcholine. Okay, now this is the structure of a synapses during nerve impulses. Please try to hold on to this image while I walk you through the processes of nerve impulses transmission along the synapse. Okay. Firstly, when an action potential arrives at the axon terminal of the presynaptic neuron, the membrane depolarizes and voltage-gated calcium ions open, allowing calcium ions to diffuse into the synaptic knob. The influx of calcium ion causes the synaptic vesicles to move inwards towards the presynaptic membrane. The vesicle then fuse with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft via exocytosis. The neurotransmitter diffuse across the synaptic cleft and bind to the specific receptor molecules on the postsynaptic membrane. This binding causes associated sodium ion channels onto the postsynaptic membrane to open, allowing sodium ions to diffuse into the postsynaptic membrane. It is, if enough, neurotransmitter molecules bind to the receptors and an action potential is generated in the postsynaptic neuron. Now we discuss about the role of neurotransmitters. Neurotransmitters are essential for transmitting nerve impulses across the synapse. Neurotransmitters can either be excitatory or inhibitory. Excitatory neurotransmitters like acetylcholine and glutamate causes the opening of sodium ion channels, leading to depolarization of the postsynaptic membrane and increasing the likelihood of an action potential. Inhibitory neurotransmitters causes the opening of potassium chloride ion channels, resulting in hyperpolarization of the postsynaptic membrane, which makes it more difficult for an action potential to occur. Neurotransmitters must be removed from the synaptic cleft to prevent continuous stimulation of the postsynaptic neuron. This is often done through enzymatic breakdown or reuptake by the presynaptic neuron. Next is acetylcholine. The common neurotransmitter of acetylcholine is a common neurotransmitter in the nervous system. It is used at neuromuscular junctions, in the parasympathetic nervous system, and in certain parts of the brain. Acetylcholine typically has an excitatory effect on the postsynaptic membrane by opening sodium ion channels down. After acetylcholine has stimulated the postsynaptic membrane, it is broken down by enzymes acetylcholinesterase into acetate and choline. The choline is then reabsorbed by the presynaptic membrane and recycled. Synapsis ensures that nerve impulses travel in one direction because neurotransmitters are only released from the presynaptic side and the receptors are only on the postsynaptic side. One neuron can connect to multiple other neurons at a synapse, allowing nerve signals to be sent in different directions from a singular presynaptic neuron. Now we discuss about the effects of drugs. And then we will look at some of the drugs such as nicotine, lidocaine, cobra venom, alpha toxin, L-dopa in the treatment of Parkinson's disease, and the action of MDMA on our nervous impulse transmission. So firstly, let's discuss about how drugs can affect the nerve impulse transmission. Drugs mainly work by two ways. First is drugs can increase the response. Second is drugs can decrease the response. When we're talking about drugs increasing the response, we specifically say that drugs increases the amount of neurotransmitters that is being synthesized, increases the release of neurotransmitters from the vesicle of the presynaptic membrane, binds to the postsynaptic receptors and activates them or increases the effect of the normal neurotransmitters. This and prevents degradation of neurotransmitters by enzymes or prevents reuptake into the presynaptic knot so that the neurotransmitters can remain in the synaptic cleft for a prolonged period of time so that more action potential can be triggered in the postsynaptic membrane. Drugs can decrease the response of the nerve impulse transmission by the following ways. Firstly, it blocks the synthesis of neurotransmitters. So there is no synthesis of neurotransmitters. So no neurotransmitters can be synthesized and sent to the postsynaptic membrane or released into the synaptic cleft. And this also causes neurotransmitters to leak from vesicles and be destroyed by enzymes. Drugs can also prevent the release of neurotransmitters from vesicles or block the receptors and prevents neurotransmitters from binding. These are some of the ways in which drugs affect the nerve impulse transmission. Okay, now let's talk about the specific examples of drug action. So first is nicotine. Nicotine is found in tobacco, has multiple effects on synaptic transmission. It mimics acetylcholine by binding to the nicotinic acetylcholine receptors on the postsynaptic neuron, initiating an action potential. It causes a prolonged period of unresponsiveness of the receptors after stimulation. It stimulates the release of dopamine, which is associated with pleasure and reward, thus making nicotine addictive. It increases heart rate and blood pressure. Next is lidocaine. Lidocaine is a local anesthetic used to numb areas of the body and regulate heartbeats. It works by blocking voltage gated sodium channels in the postsynaptic membrane. This prevents the influx of sodium ions that are needed for depolarization, thus preventing the generation of an action potential and inhibiting the transmission of nerve impulses. Next is cobra venom or alpha cobra toxin. Cobra venom is a potent toxin that affects neuromuscular junctions. It binds to acetylcholine receptors in the postsynaptic membrane, preventing acetylcholine from binding. This blocks the influx of sodium ions and inhibits the generation of action potential, leading to muscle paralysis. It can cause paralysis of breathing muscles, which can be fatal, though in small quantities it can also be used as muscle relaxant. L-DOPA. L-DOPA is used to treat Parkinson's disease, a condition characterized by a deficiency of dopamine. L-DOPA is a precursor to dopamine and can cross the blood-brain barrier, which dopamine itself cannot. Once in the brain, it is converted to dopamine by the enzyme dopa decarboxylase, which increases dopamine levels in the brain. This helps to reduce, this helps to increase transmission of nerve impulses in areas of the brain that control movement, thus improving motor control and reducing symptoms of Parkinson's disease. Next is MDMA, ecstasy. MDMA is a recreational drug that affects multiple neurotransmitters, particularly serotonin. It inhibits the reuptake of serotonin by binding to the proteins that normally enable serotonin reuptake on the presynaptic membrane, leading to increased serotonin levels in the brain. It also triggers the release of more serotonin from presynaptic neurons, further increasing serotonin concentrations. And the increased serotonin levels can lead to feelings of euphoria and enhanced touch and bodily sensation. Next is how the nervous systems of organisms that detect stimuli with reference to rods in the retina of mammals, the roles of rhodopsin, opsin, retinal, sodium ions, cation channels, and hyperpolarization of rods in forming action potential in the optic neurons. Now, the nervous system uses specialized receptor cells to detect stimuli, which then generate electrical impulses in sensory neurons. In the retina of the mammals, rod cells are photoreceptors that detect light. Particularly in low light conditions, the process by which rod cells generate action potential in optic neurons involve a series of chemical and electrical changes. Rhodopsin's role. Rod cells contain a light sensitive pigment called rhodopsin. The rhodopsin is made up of two components, opsin, a protein, and retinal, a light absorbing molecule derivative from vitamin A. Retinal can exist in two forms, cis and trans. When light hits rhodopsin, the cis-retinal converts to trans-retinal, causing a change in the shape of the rhodopsin molecule. This change puts a strain on the bond between the opsin and retinal, leading to the breakdown of rhodopsin into opsin and retinal in a process known as bleaching. Now, the role of sodium ions and cations channels. Normally, in the dark, rod cells' membrane are permeable to sodium ions. Sodium ions move into the rod cells through sodium ion channels down a concentration gradient, while the sodium potassium pump actively moves them out. This movement of sodium into the cell results in a slightly negative charge inside the rod cell relative to the outside, maintaining a state of depolarization. When rhodopsin is bleached, it triggers a cascade of reaction that leads to the closing of sodium ion channels in the rod cell membrane. The sodium potassium pump continues to actively transport sodium ions out of the cell because the sodium channels are closed. These ions cannot re enter. Now, next is the hyperpolarization of rod cells. The closure of sodium ion channels means that fewer sodium ions can enter the cell as sodium ions are moved and cannot re enter. The inside of the rod cell becomes more negative than at rest, resulting in hyperpolarization. This change in membrane potential, the hyperpolarization results in generation of potential known as generator potential. When the rod cell is depolarized in dark, it releases an inhibitory neurotransmitter that prevents action potential generation, the bipolar neurons. When the cell rod cell is hyperpolarized in light, the inhibitory neurotransmitters is not released. Action potential in optic neurons. The hyperpolarization of rod cells causes the rod cell to stop releasing the inhibitory neurotransmitter, which would usually prevent an action potential being generated in the neighboring bipolar neuron. This lack of inhibition allows an action potential to be generated in the bipolar neuron connected to the rod cell. If the generator potential is strong enough, the bipolar neuron sends an impulse through the optic nerve to the brain. If several rod cells are stimulated at once, the generator potential can converge to generate an action potential in the bipolar neuron. This is the all or nothing law, where an action potential generation follows an all or nothing mechanism, meaning that the generator potential reaches the threshold of the sensory neuron, then an action potential will be generated in that specific neuron. In the dark, now here are some additional points that you need to remember that in the dark, rhodopsin is resynthesized with transretinal being converted back into cisretinal, which then joins opsin using ATP from the mitochondria. Then moving from a dark to a light area, the visual pigments are bleached and it takes time for rhodopsin to be resynthesized, resulting in a temporary reduction in vision. Rod cells provide low quality images for black and white vision in low light conditions, whereas cones provide high quality color vision in bright light. The intensity of the light stimulus is detected by the brain by the frequency of action potentials and the number of neurons which carry these action potentials. Let's now look at how the pupil dilates and contracts. In bright light, photoreceptors in the retina detects the high light intensity. A sensory neuron then sends electrical impulses to the central nervous system. The sensory neuron synapses with a relay neuron in the brain. The relay neuron in turn synapses with the motor neuron. The motor neuron carries an impulse to the circular muscles in the iris, which acts as the effector. The circular muscles contract, causing the pupil to constrict and reducing the amount of light entering the eye. At the same time, the radial muscles relax. Negative feedback. This is the part of a negative feedback loop, preventing damage to the eye due to excessive light. And in dim light, photoreceptors in the retina detected the low light intensity. A sensory neuron sends electrical impulses to the CNS. The sensory neuron synapses with a relay neuron in the brain. The relay neuron synapses with a motor neuron. The motor neuron carries an impulse to the radial muscles in the iris. The radial muscles contract, causing the pupil to dilate, which allows more light to enter the eye. Simultaneously, the circular muscles relax. The muscle of the iris are two types, circular muscles and radial muscles. These circular muscles are arranged in concentric rings around the pupil. When they contract, they constrict the pupil. Radial muscles extend upward from the pupil like the spokes of a wheel. When they contract, they dilate the pupil. The circular and radial muscles work antagonously. When one set contracts, the other relaxes. What is meant by the term habituation? Habituation is a form of learning where an organism reduces its response to repeated harmless stimulus. This process allows an animal to conserve energy by not responding to non threatening stimuli, thus saving resources for situations that are actually dangerous or require a response. The fundamental aspect of habituation is a decrease in the response to a stimulus after repeated exposure. So, habituation typically occurs with stimuli which does not have any negative consequences. This allows the organism to ignore things that are not harmful or not respond to things which may not cause them any harm in order to conserve more energy. Habituation helps animals to save energy by not reacting to every stimulus, thus making them more efficient in their environment. The physiological mechanism behind habituation involves changes in synaptic transmission. During habituation, there is a reduced influx of calcium ions into the presynaptic neuron upon the arrival of a nerve impulse. This reduced calcium ion influx leads to the release of fewer neurotransmitters in the synaptic cleft. Consequently, fewer neurotransmitters bind to receptors on the postsynaptic membrane, reducing the likelihood of an action potential being generated in the postsynaptic neuron. If the postsynaptic neuron does not reach the threshold for action potential generation, the nerve impulse does not continue and the organism does not respond to the stimulus. Habituation is not permanent. If the stimulus changes, the organism can regain its sensitivity and respond again. So, here are below some examples of habituation, such as humans no longer noticing a new smell or sound after a period of exposure, or wild animal losing their fear of humans after regular non harmful contact, or animals learning not to be alarmed by the presence of non predatory species. We have reached the end of the video. Thank you so much for watching. I hope you found this video helpful. If you have any comments or any questions, feel free to ask me in the comment section below. See you next time.